There is a fentanyl pandemic out there, and fentanyl is an opioid that is very toxic. So its manufacture is very straightforward, more so than cocaine and other drugs out there that are commonly available in the streets. Something that makes it an even scarier situation with fentanyl is that it's very powerful, meaning that a few grains amounting to maybe like a few grains of salt, by comparison by volume, that could be lethal for anyone who takes it.
There's not a lot of choices out there for treatment. Naloxone is the one that's probably used the most, where naloxone is more of a reactionary treatment where you've already been exposed and then you're trying to compete with the fentanyl. It's effective, but it's also very short-lived.
There was a call from the Department of Homeland Security back in 2011, 2012, I want to say. They were looking at the fentanyls as a potential target for development of better antidotes for it, at least at the Forensic Science Center at the lab. It made us aware of the fentanyl problem. We decided to see if we can use some type of specific technology to develop a medical countermeasure against it. And a medical countermeasure is defined as something that goes into your body to protect you from the effects of the drug or the toxic substance before it reaches its target. We were able to find a molecule that can do that. It's called subetadex.
And one thing that we're also trying to do is have this act prophylactically, where you can give someone the drug before they're even actually exposed. So when they go into a situation, they're not going to have the effects of the opioid. This can be used for all different types of exposure scenarios, for overdose treatment, as well as for first responders and military personnel that might come into a situation where there has been an exposure. So developing this drug that can be used both prophylactically and therapeutically would be a great advantage.
Back in, I want to say like 2015, more or less, we were testing a number of very similar molecules initially in the project, and we're using an experimental technique known as nuclear magnetic resonance. What basically tells you is whether or not a fentanyl is binding the sugar that you're testing. And after tens of failures and finding a molecule that could bind fentanyl, we came across one that was a predecessor to subetadex. We found that by NMR, by the nuclear magnetic resonance, that it was binding fentanyl quite well, and it was not letting go of it. That was a big moment. We were able to identify already a candidate, a platform. When you find a compound that does something already, you get excited because you know as a chemist you can come in and you can modify that compound to make it even better.
In collaboration with others at the lab, we used computational chemistry as well as chemical synthesis, as well as biochemistry and in vivo testing to adapt these existing molecules so they would interact with fentanyl to deactivate it or neutralize the fentanyl by making adjustments to these what are called cyclodextrin molecules. And what they do is they kind of scavenge the fentanyl once you've been exposed to it to inactivate it. So that's kind of what we did to develop this new molecule, and then we needed to test it to make sure it would actually work and make sure that it wasn't toxic as well. So we did a lot of toxicity tests of this cyclodextrin molecule to make sure it was non-toxic if we were to administer it to people, and then that came out as being, favorable, where we didn't see any toxicity. I'm a toxicologist by training, so I am interested in toxicants and drugs and things like that, and what their effects are on the body. And when this project came up, it really interested me because it is a big problem right now, the whole opioid crisis. So I thought, with my skill set, I could really contribute to this project in a positive way.
The next step on this will be to apply for what you call a pre-new drug application with the FDA. That application, what it does is that the FDA has specific regulations on how you test the drug even further. One thing that inspired this work on this specific carbohydrate molecule was a carbohydrate molecule that is out in the market. They already use it in the hospitals. It's FDA approved, and it's known as Sugamadex, and will basically follow the same steps that they did in order to seek FDA approval.
We're at a stage now that we're ready to publish our results and release them out to the public. The process that we're using to develop this is kind of the uniqueness of this whole project. By using the computations and integrating that with the chemistry and the biology, it really hasn't been done before at this scale. And I think that's where we need to really focus if we're going to continue this type of work, is using all the components together as one, which is going to speed up the process, which is a really big issue because right now, in order to get a drug to market, it takes at least 10 years because of all the testing that has to be done. But if we can do more of the upfront testing in the computational realm, we can reduce a lot of the biological testing that has to be done to decrease what we call time to market. We hope that this drug will lead to better outcomes for people who are exposed. And by developing these type of drugs, it puts more in your tool bag to treat these exposures, and getting this to the point where we can develop it into a drug and get an FDA approval so it can get out there and help as many people as possible. I think that's where we're headed.